Okay, now let us look at seeds. So we are going to look at seeds. Then what is a seed? From a flower, from flowers we discussed that we have a male part of a flower and a female part of a flower, where we said a female part of a flower, which is called a pistil, it has 3 parts. It has this one, which is called the stigma. Then we have the style. Then again we have the ovary. Then inside the ovary we are having the ovules. So these ones are the ovules. These are the ovules. And then we have the male part of a flower, which is called a stamen. It has the anther and the filament stalk. Then this anther produces the pollen grains, which land on the stigma, and they develop into the pollen tube. So they grow into the pollen tube. Then the pollen tubes are going to be carried down the ovary through what we call the style. On reaching the ovary, they are going to meet with the ovules. This one is what we call fertilization. So after the process of fertilization, this one is the ovules are going to turn into what we call seeds, and the ovary is going to become what we call a fruit. Therefore, a seed is a fertilized ovule. A seed is a fertilized ovule. And again, these seeds, for them, one of the characteristics they do have, they have only one scar. So anything to be called a seed, it has to come from the ovule, and it has only one scar, and the scar of this seed is called the hilum. So now let us look at the types of seeds. Then these seeds are classified into 2 types. 1, we have what we call the dicotyledonous seeds, and 2, we have what we call monocotyledonous seeds. And then let us start with what we call the dicotyledonous seeds. So dicotyledonous seeds, this word is made up of 2 words. One is di, which means 2, and the word cotyledonous or cotyledonous. So this word cotyledonous comes from the word cotyledon. So meaning that dicotyledonous seeds are seeds with 2 cotyledons. So these are the seeds with 2 cotyledons. Dicotyledonous seeds are seeds with 2 cotyledons. Then let us take examples. Examples of seeds with the 2 cotyledons. 1, we have the beans here. So we have the peas, then we have the soya, and we have the ground nuts. So these are the examples of seeds that have 2 cotyledons. Remember, we said dicotyledonous seeds are seeds with the 2 cotyledons. Then let us now look at the structure of a bean seed. The structure of a bean seed, and label the parts that are there. Now when you look at this bean seed here, it is showing us the external parts, whereby this outer colored part is called the tester or the seed coat. Then here we have the position of the plumule, and here we have the position of the radicle. Then here we are having the hilum. Then at this point here we are having a small opening which will let in water and oxygen during the process of germination, as we are going to discuss further. So now here we are going to look at the internal parts of a bean seed. So what I'm drawing here, it is the outer part, this one which we call the seed coat. So this one is the seed coat, and it's very important. So the importance of a seed coat is to protect the inner parts of a seed. So it protects the inner parts of a seed from mechanical damage and from being eaten by the insects that are found inside the soil, in case this one has been put inside the soil. Then up here we are going to have what we call the plumule. So this part here is called the plumule, and it is this plumule which is going to develop into the shoot system. So remember, from our classification where we started from, we discussed the plumule, the shoot system. Therefore, this shoot system comes from a part of a seed called the plumule. Then below here we are having also another part, and this part is called the radicle. So this radicle is the one which is going to give us what we call the root system during the process of germination. Then this entire part here is what we call the cotyledon. So we said the bean seeds, they have two cotyledons. So this one is the cotyledon, one cotyledon. If now we draw here another one here, it's going to give us the second cotyledon. And then at the center here we are having two things. One, we have this scar which is called the hilum, and the importance of the hilum, it is to attach the seed onto a pod. Then here this one is what we call the micropyle. So this part here, let us name this one together. This one is the tester, this one is the cotyledon, this one is the radicle. This one, this small attachment here, this small opening is called the micropyle. This one is the hilum, and this one is what we call the plumule. So the plumule develops into a shoot system, the radicle gives us the root system, then the cotyledon, its work is to store and supply food to the embryo. So when I talk of the embryo, when we are discussing the seeds here, this word embryo means the collection of the combination of the plumule plus the radicle. So instead of saying plumule and the radicle, we use one term that is called the embryo. So this embryo grows or develops into a seedling, as a seedling being the young plant. Then this micropyle, it is a tiny opening, it is micro, so it is a tiny opening through which water and oxygen pass to enter inside the bean seed during the process of germination. So this is a bean seed. So now let us look at so now that has been the dicotyledonous seeds. Then let us look at what we call monocotyledonous seeds. Remember we said mono means one, this one means one, then cotyledonous means cotyledon. Therefore monocotyledonous seeds are seeds with only one cotyledon. So please, I urge you, after here you write the notes that are sent through the WhatsApp number. Now the best examples of the monocots or the monocotyledonous seeds, one, we are having what we are seeing here. We are having the maize, we have the maize grain, two, we have the wheat, three, we have the rice, then we have the sorghum, we have the millet, so we have the oats, and also have what we call the barley. So all these ones are examples of monocotyledonous seeds, the seeds with only one cotyledon. So now here let us look at the diagram showing a maize grain. So I'm drawing this maize grain just like this one, then I give it just this part here. Then it has two linings, so I'm drawing this lining here. Then I also bring this lining here, just like this. So now this outer lining, this one, it is what we call the seed coat, as we've seen in the maize grain. And once you don't call this one a seed coat, it is also called the tester. And we say that this tester or the seed coat, it provides protection to the inner parts of a maize grain. And then it has this part here, the upper part, which we call the style scar. So this one is the style scar. Then it has this lower part here, which you call the stalk scar. This one is the stalk scar, and the importance or the function of this stalk scar, it is to hold, it holds the grain, onto the what, the cob. So like the hilum holding the maize, the bean seed onto the pod, then the stalk scar, it holds the grain onto the cob. Then this entire part here, it is divided into two portions here. So we are having this entire part here, then this one, it's a cotyledon, so it has only one cotyledon. Then this larger part, this largest internal part here, it is called the endosperm. So this one is the endosperm. So it has the style scar, the endosperm, the cotyledon, the seed coat or the tester, then the stalk scar. Then inside the cotyledon here, we are having two parts. One, we are having this one, the upper part, and this one in the middle. So this one is what we are going to call the epicotyle. this one is what we call the epicotyle. Then this one next to the epicotyle, it is what we call the plumule. And remember we said the plumule develops into the shoot system. Then down here we are having also two. Then this outer one, the last one down here, it is called the coryhaza. This one, it is the coryhaza, or it is called the radical sheath. This one is the radical sheath, and if you don't call this one the radical sheath, you can call it the hypocotyle. This one is the hypocotyle. Then this one here, it is our radical. So this one is the radical. Then with a combination of this plumule and this radical, it is called the the embryo. Therefore, let us name this seed together. This is the stalk, this one is the endosperm, then this is the stalk, then this is the radical sheath or the hypocotyle, then we have the radical, then we have the cotyledon, then we have the plumule, and what we call the epicotyle. Then we are going to take some brief notes about the maize grain. One, this maize grain is called aferiot. So why do we call this maize grain aferiot? It is called aferiot because it has two scars. It has two scars. So it resembles a fruit because it has two scars, and all fruits have two scars. Remember we said what we call a seed, it has to be with only one scar, but we are seeing this maize grain having two scars. So it has the style scar and the stalk scar. Number two, this maize grain stores its food in the endosperm. So it stores food in the endosperm. So the endosperm stores food for the grain, whereas the beans or the dicots, they store their food in what we call the cotyledon. Then its cotyledon here does not store food. Note number three, the cotyledon of a maize grain does not store food, but it supplies food. It supplies food to the embryo. It supplies food to the embryo. Remember we said embryo, this is the combination of the plumule and the radical. So this cotyledon of a maize grain does not store food. It supplies food to the embryo, that is the plumule and the radical. So it absorbs food from the endosperm and supplies it to the embryo. Whereas now and the cotyledon of a bean seed, it does it because there are two cotyledons. So it does or they carry out two functions. One, they store, two, they supply. So the cotyledons of the bean seeds, they store and supply food to the embryo, whereas this cotyledon of the maize grain, it only absorbs food from the endosperm and supplies it to the embryo. Remember we've said the embryo, that is the collection of plumule, or the one word given to the plumule and the radical. I think that one's made clear. So actually that's a common question. They will ask you to give the difference between the cotyledon of a maize grain and that of maybe a millet or wheat or maize grain. So the difference is that this one stores food, stores and supplies food to the embryo, whereas this for the maize grain, it only supplies food to the embryo after absorbing it from the endosperm. So all monocotyledonous seeds, they store their food in the endosperm. Now let us compare what we have named here with this one. So this entire part you are seeing here is what we have called the endosperm storing food. Then this one you are seeing here, it is our cotyledon. So it will absorb food from the cotyledon and supplies it to the embryo. Now this one we are seeing, it is what you call the epicotyle or the coleoptile. Then down here is what you are seeing is what we call the hypocotyle or the coryhaza. Then this one is the radical and this one is the plumule. So this one is the plumule, this one is the radical, then this one you are seeing is the radical sheath or the coryhaza or the hypocotyle. Then this is the radical sheath or what you call the epicotyle. So this part you are seeing here, this is the stalk scar. So this is the stalk scar holding this grain onto a cob. Then up here we are having our style scar. So this is the style scar, then the endosperm, then the tester, we have the stalk scar, we have the hypocotyle, we have the radicle growing into the root system, we are having the plumule growing into the shoot system, we are having what we call the epicotyle. So this one is showing us the diagram of a maize grain, and we say it is also called a fruit simply because it has two scars. So now let us move on and see what to the soil. So when these seeds are put into the soil, they are going to undergo what we call germination. That is what we call seed germination. Therefore, a germination is the development of a seed. Germination is the development of a seed into a seedling. Is the development of a seed into a what? A seedling. Then this word seedling, it means a younger plant. So a seedling is a younger what? A plant coming from a seed. But now these seeds to develop into seedlings, they have to be under, or they have to obtain, or they have to get what we call favorable conditions. Under favorable conditions. Therefore, germination is the development of a seed into a seedling under favorable conditions. Now, let us look at these conditions, the things these seeds need in order to germinate very well. 1. These seeds, they need oxygen. The seeds, once they are put in soil, they need what we call moisture, not water, because water can make them rot. So moisture, reasonable amount of water. And 3, they also need what we call the warmth. So these are the 3 conditions needed by seeds to do what? To germinate. All the conditions for seed germination. Seeds need oxygen, they need moisture, and also they need what we call warmth. Now let us see, why do these seeds need oxygen? This oxygen is very important. These seeds need oxygen. Why? So the role played by oxygen during the process of germination is that it carries out respiration. So respiration, as we are going to discuss later on, we are going to understand that the process by which we are obtaining energy. Living organisms are obtaining energy. So when these seeds are also germinating, they need energy in order to establish new organs and new parts in the roots, the root hairs, the leaves, the stems, and so on. So oxygen is very important in order to carry out the process of respiration during the process of germination. Then again, why do these seeds also need warmth? Warmth, that is the optimum temperature. So the temperature must be optimum in order to do what? To activate the enzymes, to wake up the enzymes, to make the enzymes active. But don't say to wake up enzymes, no, we use the scientific term activate. So optimum temperature, that is warmth, it is needed in order to activate their enzymes, to make them active in order to break down the food during the process of respiration, which was stored in the cotyledon, to provide the organs, the embryo, with energy to establish new organs. So the optimum temperature or the warmth is needed to activate the enzymes during the process of germination. So these enzymes were discussed fully in primary 5. So now let us look at another condition, that is moisture. Why do these seeds need moisture? 1, this moisture it softens the seed coat. Moisture softens the seed coat. It softens the seed coat. Number 2, this moisture also dissolves the starch, or it hydrolyzes, but let us use this word, dissolves the starch that has been stored into or in the endosperm or in the cotyledon. So water has 2 benefits: 1, to soften the seed coat, 2, to dissolve the starch. Then this water, if you remember the bean seed, this water, the bean seed, it has a small opening. Then this small opening, which we call the micropyle. So it is through this small opening where water and the oxygen pass. The process by which this water is entering inside the bean seed here, it's called imbibition. So that is water uptake through the micropyle. Now During this process of germination, one, the seed has to take in water. It takes in water from the soil. Then the seed coat will swell. After swelling, then it will burst. Then after bursting, the first part to come out during the process of germination, that is the radicle. So the radicle comes out first. Why? To absorb water from the soil and also to anchor or to hold the seedling into the soil family, to provide support, because once now the roots aren't there, then it can be taken away, it can be blown by wind. So it's the radicle to come out first, one, to absorb water, and two, to anchor the seed or to hold the seed family into the soil. Then after that, then the shoot system is now going to follow, is going to come out of the soil. Then we are going to look at two types of germination. So we have types of germination, and these types of germination, they are only two. One, we have what we call the epigeal germination, and two, we are having what we call the hypogeal germination. Then epigeal germination, here we are seeing the cotyledons coming out of the soil. Two, the hypogeal germination, we are seeing all the cotyledons remain inside the soil. Now I'm going to illustrate here, I'm going to draw here a simple diagram which I'm going to use to explain this germination. So let us assume we are having our seedling with the leaflets here, then it has this one, then it has these things here, then it continues, then it has the roots here, it is in the soil. Now when we look at this small diagram I've drawn, these are the leaves, but these are not leaves. These ones are called the cotyledons. Then this upper part, the part above the cotyledons, it is called the epicotyle. Then this lower one is not called the stem, it is called the hypocotyle. Then I want you to get me clearly here. Now if the seed is germinating, now if the hypocotyle comes out fast, grows faster than the epicotyle, then the cotyledons are going to come out of the soil. Now if it is the epicotyle to come out fast, then it means that the cotyledons are going to be left down, then it is going to become the hypogeal germination. So to understand this one, let us now observe this video here and see what is taking place. So now here we are having our bean seed, so it is in the soil, then the cotyledon, the radicle is coming out fast. So as it is coming out fast, it is, as you are seeing here, it has absorbed water fine, then it is coming out, it is now developing, establishing the root hairs. Then observe here. Then observe, so this part here is what we call the hypocotyle. So it is this hypocotyle growing faster because the epicotyle is inside here, the cotyledons. So this one has come, it has grown, or it is growing faster than the epicotyle. Therefore, observe again. So as you are seeing, this hypocotyle, it is pushing up at this. So as it has been pushed out, so as it has been pushed out, then the cotyledons have come together with the seedling. So so this is our epicotyle, then this is our hypocotyle, and these are the cotyledons. Thus this one making what we call the epigeal germination. Let us observe it one more time. So here, this is a seed, this is our radicle. I said the radicle comes out first in order to do what? To absorb water and to anchor the seedling into the cell family. Then this is what we call our hypocotyle. So the hypocotyle is growing faster than the epicotyle because it is inside there between the two leaflets, the plumule. Then as it is growing faster, it is now pushing the upper part up. Thus it is taking out the cotyledons outside. Then this one is making what we call the epigeal germination, whereby the cotyledons come outside the soil. Why? Because the hypocotyle here grows faster than the epicotyle, as you have observed or as you are seeing there. You have seen that. Now let us also observe what we call hypogel germination here. Remember we said here the cotyledon remains inside the soil. It remains inside the soil simply because the epicotyle, as we've been seeing, it will now grow faster. So as it comes out fast, it will leave the cotyledon and other parts down the soil. Let us observe this here. So we are observing now here our radicle is coming out. As this it has to come out fast always. Then observe here, as we are seeing this, then here it is now the epicotyle coming out faster, growing faster than the hypocotyle again. So as it is coming out fast, then the hypocotyle will remain down, then the radicals are also going to remain down the soil. Let us observe it for one more time. Then as you are seeing, the radicle is there coming out, developing the root hairs. The root hairs are there, the lateral roots making the fibrous system. This is the maize grain or wheat or barley. Then the hypocotyle has come out. Then so as let me rewind it for one more time. You see this also, this part. So as this coming out, it's going to come out. This hypocotyle, it's now the epicotyle coming out, growing faster than the hypocotyle. That's going to leave the cotyledon inside the cell. Then as this coming out, it's going to come out with a sheath, a membrane, and that membrane is what we called the coleoptile, as we named the maize grain. So that coleoptile, it is to protect the leaves, the shoot system, the plumule from getting damaged, because when you look at this soil, we are having stones, the insects are there, so they can cause damage onto the leaves. Thus no development of a proper seedling. Let us observe this. So this one is enclosed, so observe it as it is going. So it is in a coleoptile. Then as it grows, still it is in the coleoptile. So as it grows, as they are coming out, they are coming when they are covered. So until they come out of the soil, all of them, then this is still at the here it is opening. I think you've seen now the coleoptile has opened to let the leaves come out now to be exposed and to sunlight, as you are seeing here. So this is the coleoptile, then the leaves are getting exposed to sunlight. Continue. So we are now going to look at our activity. So but before getting into the activity, there are some notes I want us to make. Note 1, the coleoptile, this coleoptile, it is that it covers the shoot of the monocot seed when it is germinating, in order to provide protection. This one provides protection to the plumule or to the leaves as we are getting the hypogel germination. Then with the epigel germination, we said the hypocotyle grows faster, so it develops fast. And lastly, we have looked at the hypogel. Then the hypogel germination, here the epicotyle comes out fast. Thus here the cotyledon remains inside the soil, as you've seen. Then here the cotyledon comes out of the soil. So I urge you, before doing the activity, to first copy the notes that are attached onto this video. Then you answer the activity, or you write the activity. Our number 1, define the term germination. Define the term germination. Number 2, mention the two types of germination. The two types of germination. Number 3, which part of a seed develops fast during germination. Number 4, identify the two types of seeds. Number 5, state the difference. State the difference between epigel and hypogel germination. Hypogel germination. Number 6, where do the following seeds store their food? Roman 1, maize. Roman 2, beans. Why is a maize grain? Why is a maize grain called a fruit? Number 8, give the function of the following parts. Of a seed. Roman 1, seed coat. Roman 2, tester. Roman 3, micropyle. Roman 4, cotyledon. Number 9, draw and label all the parts of a maize grain. Number 10, state the difference in function of a cotyledon of a maize grain and a bean seed.